Hello everybody. We were talking about the auditory processing, and today I'll talk about the auditory scene analysis. As you know that using our auditory system we can perceive the world around us. We can sense the things happening around us. Although the ability of auditory scene analysis is pretty weak in human auditory system or brain. As you know that some creatures have much higher ability to do the auditory scene analysis or sound localization. Human has some limitations. As you know that a blind person cannot do a lot of things with the help of the auditory system. Um, However, we can use different techniques to localize the sound, and one is called the auditory stream segregation. Auditory stream segregation is the ability to divide the auditory world into separate auditory objects. That means, it is a perceptual organization of a complex acoustic signal into separate auditory events for which each stream is heard as a separate event. So you know that a lot of things are happening at the same time around us, right? How you can get the complete picture. The good example could be like, when a blind person enters into a restaurant, he uses his auditory system to do the auditory stream segregation. That means to perceive the things happening inside the restaurant. So you know that his auditory system will pick up the signals, multiple auditory signals, and will group them into separate streams. For example, from the kitchen area, you know that the sound comes from the kitchen area, or like, somebody is frying something, and someone is like, putting something, using the spoons and mixing something. Those kind of sounds will come from the kitchen area, right? So you see that multiple things are happening inside the kitchen, and your auditory system will group them, all those things together, and will give you the picture of the kitchen. At the same time, the area where the people are sitting and eating and they're talking, chatting, different types of sounds are coming from that area. Although multiple sounds are coming from the sitting area or eating area, but all those are grouped together in the auditory system. Your auditory system is capable to do that. Okay, group similar type of sounds together, and that is the stream segregation. Okay, another good example: if you close your eyes and someone drops a glass on the floor, by listening the sound you can tell if the glass broke or did not break. How? If it did not break, then you will listen the sound, that one unique sound coming from a single piece, that is the glass. And you will also listen the bouncing sound, which is different if the glass breaks into multiple small pieces. Because if the glass breaks into multiple small pieces, then each piece will produce separate sound at different onsets, right? And at different frequencies because of the size of those pieces. is different. Okay, so your auditory system can analyze those sounds to give you the perception about the event. In this case, onset of sound is important, right? If the glass is intact, it's a single onset, right? And you will hear the bounce, right? And gradually it will, time will get shorter. Okay, and if it gets break, then you will see multiple pieces and sounds are coming from multiple pieces. So you'll see many, at many different frequencies. So each piece will have its own onset of sound. Another good example is if you present two tones in close frequency differences. That means the two tones are presented, but the frequencies are pretty close to each other. Then if you present them like this is one tone right here, and this way is frequency. So this is slightly higher frequency, this one, and these are the tones. So you see, this is one frequency, this is another frequency. So in two slightly different frequencies you present two tones, and if you present them very fast and in rapid succession, then what will happen, that you will perceive a single tone, a single warbling stream. Okay, in this case. However, if you present two series of tones at two very different frequencies, then two separate streams or tones will be heard. Okay, so you see this one, first you present this low, then this high, then low, then high, low, high, but the difference of frequency is very small, okay, close to each other. Here the frequency difference is big, and first you present this one, then this one, then this in rapid succession. Then you will hear, you will hear two separate streams. Okay, so all these examples tell us that the auditory system can segregate or separate the sources of sounds based on multiple cues, onset, or frequency presentation. Those are important cues for your auditory system to segregate or separate the sources of sounds. Another example, like if you enter into a park, inside that park many events are happening, right? If you are blind, then you will use those cues, and you will be able to tell that what kind of ride is in which areas. So you can do that. Or if you enter into a forest where in one side you have the water, and in the other side you have the bushes, you can tell by listening the sound from the bushes. You will hear the sound of wind and the trees, leaves, or even the birds are singing, right? And one type, although multiple sounds, but your auditory system will group them together and will picture all those as one image. And in another side you will hear the sound of water and ducks, or those kind of sounds will give you the picture of that lake or water. So our auditory system can do that. Continuity effect. This is something interesting property of the auditory system. your auditory system can restore the perception. That is called the perceptual restoration. When a sound is uh, deleted and replaced with a loud noise, then listeners will hear the sound as if it had continued through the noise. You won't feel that interruption or discontinuation of that sound. And this is true when a tone maintains the same frequency and when the tone steadily increases or decreases in frequency. So here you see two situations. This is brief presentation of noise. This is the sound, so frequency here, and this is same frequency, right? So you present the sound same frequency and you are listening the sound, and then you briefly present noise. This dark bar is showing the noise. However, after that, if you ask the subject that did you hear any interruption, or did you miss anything, the subject will say no. So that is one interesting property of your auditory system called the perceptual restoration. Your auditory system can continue to listen that sound by ignoring the noise. This is also true if the sound not in same frequency, steady increase of the sound or decrease. So if you go steadily upwards or downwards this way, either way, and during that steady increase or decrease, if you briefly present the noise, and after that if you ask the subject did you feel anything, the subject will say no. He will continue to listen the sound. Okay, and that is something interesting property of your auditory system and helps in auditory scene analysis by ignoring the disruption. And a very interesting experiment was performed to see how the neurons respond during this noise presentation. And one very interesting experiment which was performed by Sugita, a Japanese scientist, in 1997. Interestingly, during this time I was in Japan. Okay, and he recorded from primary auditory cortex or A1, the neurons, and what he found that during this time when the stimulus was presented, of course the same frequency, if this is like 40 kilohertz for example, you will see 40 kilohertz neurons are firing here. So these are the neurons, will increase the firing rate, right? So that is normal. These neurons will be stimulated by that sound. And what he noticed or found that even when the noise was presented, these neurons continued firing. So this is something that neurons remain active during that period. Interesting thing is that when he reduced the noise and the noise level was low, the neuron lowered the firing. So during this period, the noise must be strong. Continue to listen that sound. This is another interesting perceptual restoration effect. The subject, this is the spectrogram, so you can record the frequency and this way time. So this is time and this is frequency. This is called the spectrogram, and the subject listened a sentence. The mailman brought a letter. Okay, so this is the spectrogram of that sentence. And then what the experimenters did, they in this case, listeners have great difficulty understanding the sentence when intervals are removed and replaced with silence. So during this time, the subject did not hear anything. So, sentence started, then silence, then rest part, then silence, then rest part, then silence. So, this is one situation. In another situation, instead of silence, they presented noise. So what they found after they performed this experiment, the subject was unable to tell what was that sentence. However, when the noise was presented instead of silence, after that the subject was able to tell the whole sentence. Okay, so when the similar intervals are repeated by noise, it becomes easier to understand the sentence. Okay, so that means during uh, the auditory perception, if a noise is presented, uh, that does not disrupt the perception a lot. However, silence can disrupt the perception, and that is something interesting, that in presence of noise, your auditory system can still recover or continue perceiving the signal. So this tells that adding noise improves perception or comprehension. Okay, so now, as you know, that auditory scene analysis, what human brain does is relatively simple. Although it is not simple, it is very complicated, as I mentioned before. It's more complicated than the visual, because we have no way to stop the sound from entering into the ears. Visual has the ability, you can close the eyes, or take away your eyes from the scene. However, if you compare the human auditory scene analysis, which is much simple compared to like bats or owls, you know that they do complex auditory scene analysis. That means they depend on their auditory system uh, to analyze the world around them, right? However, they do very differently. Bats use echolocation. Bats produce pulse sound, and that sound hits the objects and returns into the bat's ear, and the bat's brain process that returning sound or echo. So that is called echolocation or biosonar. Uh, However, owls do auditory scene analysis differently. It's by sound localization, which we human also do, but our sound localization is uh, very poor compared to the owl's sound localization. The physical structure of the owl has some advantages over uh, the other creatures. And if you see the head part of the owl, um, it is very interesting. The face looks like a dish, kind of round, and the feathers in the head absorbs the sound. Okay, so it works like a dish to capture the sound. So that's why the owl will look straight towards the sound source. So its face can capture the sound and then funnels the sound into the ears. And the ears of, if you see the ear canals of the owl's head, um, they are not exactly the same level. One is slightly higher than the other. Okay, so vertically they are not at the same level, which also helps them to localize the sound, because if the ears are located in slightly different, um, locations vertically apart from each other, that uh, will help to localize the sound, because if this is the sound source, you know, this, since this ear is higher than this one will get the sound before this one. That means sound will arrive here slightly before than this one. Makes sense? From above. Now if the sound source is below, then this one will get before than this one. Makes sense? So even the interaural time difference is same in this case and this case, because both are in the midline. However, because of slightly different, uh, locations of the ear, one will receive the signal or sound before the other, and that uh, time difference will help the owl to localize the sound better. Makes sense? So owl can localize before leaving the perch. So you know that owl sits, for example, sitting on a tree, and here is a mice. This is the owl, and when the mice moves, that sound enters into the owl's ear, and owl's brain will calculate the exact location of the mice. Okay, and then the owl will proceed and very precisely will be able to capture the prey. Okay, and the precision is very high. Within 2 degrees owl can detect the movement within 2 degree, which is very narrow, very small, uh, uh, displacement or movement. Owl attacks sound source, okay, not the mouse. So it doesn't really know what the object is, but it will attack the sound source. Since it cannot see if the mouse is far, what is that object, but it can very precisely detect the source from where the sound is coming. And owls can localize the sound source, both vertical, that means elevation, and horizontal or in azimuth. Um, Human, you know that we localize the sound sources mostly on the horizontal plane. That means this is you, and we walk on the earth, you know that, and most of the sounds are coming from the surface of the earth. That means a car is here, or someone is walking, another person is here. So all these sources are coming from the horizontal plane at different, from different directions, but horizontal. We don't move up and down as the birds or owls or bats they do, right? They fly up and down vertically a lot. We don't do that. We always walk on the earth, the surface, same. So that's why we mostly depend on the azimuth, or our auditory system is used to detect the sound source in horizontal plane. However, birds and other flying creatures, they are both vertical and horizontal are needed. So they fly at different heights. So therefore, their sound localization is much better in elevation. As I mentioned that if you look at the face of the owl, it is like a dish. It acts or works as an antenna. Okay, and the owl will look straight. If it listens any sound, it will first look straight uh, towards that sound, and that sound waves will hit the face. Make sense? And the farts in the face are designed in a way that it will capture that sound waves, and then will pour the sound waves into the ear, from the face into the ear. Okay, and as I mentioned, the ears are asymmetric. One is slightly above the other. Okay, so here you see that this is the skeleton skull of the owl, and now you can see more clearly. This is the right ear here, and this is the left ear. So you can see that right one is above the left one. So if the sound is here, source of sound is here, this one will arrive into this ear before it arrives here. Make sense? So that interval time difference helps the owl to detect the sound source in elevation. Same thing here. If the source is below, then the left ear will receive the signal sound waves before the right. So opposite, right? So clearly owl can tell that if the sound is coming from above or below, because the neurons in the owl's auditory system can calculate that difference of arrival time, which is the interval time difference. Also in owl's brain there is a space map which is not present in the human brain, which is very helpful for the owl to detect the sound source immediately, very quickly. Another interesting thing is that in owl's wing, when the owls, after the owl localizes the sound, it will very fast will move, and when it will move towards the source, it will keep its face straight or eyes straight towards the sound location. So it will continuously receive the sound. Okay, if it turns the head away, then it will be discontinued, right? So it will keep looking straight until it captures the prey. That is one thing. Another is when the owl moves towards the prey or sound source, it moves very quietly, the sound without making any sound. And how it is possible when it moves the wings, in the wing of the owl, you will see the feathers are serrated. So that is one thing. Serrated uh, feathers can help them to move very quiet or silently, uh, uh, by decreasing the resistance in the air. Okay, also under the wing there is velvety feathers found on the wings and also legs that absorb the sound frequencies. Okay, so owl can very silently or quietly can reach the prey, so the prey will not notice that the owl is coming towards it. This is a very interesting experiment performed by a famous. He is the most famous, auditory space or localization research person. His name is Dr. Konishi. He used to work at Caltech, and I visited his lab. And he was a good friend of my mentor, Professor Suga. They both came from Japan at the same time, in around 1965 at that time, and both became very famous in auditory physiology. Dr. Konishi became very famous because of his work on sound localization, and most of his studies are on the birds and owls. Okay, so he studied speech production and sound localization. And Dr. Suga, he started to work on echolocation, and he used mostly he used bats, different species of bats. So he became famous in echolocation. Both of them became very famous scientists in the USA. And Dr. Suga used to work at WashU when I was his postdoc. Okay, so you see, at that time, those very early, studies, this is an elegant way to study the sound localization. The owl is sitting on a platform. So inside the soundproof room, owl would sit on a platform, and you implant electrode on the head of the owl, and you can record the activity of the neurons from that implanted electrodes. And this is interesting. He used a rod, like carved rod, you see, U-shaped, and that rod can move this way and this way. Okay, and he attached a speaker, okay, to that rod, and this speaker can move this way along the rod, that way. Okay, so you can move the rod forward and backward, and you can move the speaker laterally, sideways and medially. Okay, so in that way you can cover all the locations you want to put the speaker, right? So I can move this way the speaker, I can move the speaker this way, so in elevation, right? Also in azimuth, I can move this way and this way. So this allowed to present the stimulus from different azimuth and elevations in space. Okay, and he presented the sound through the speaker located at different locations in space, and recorded the neuron. Okay, so first you just use the electrode and record the activity of the neuron, like this is the spontaneous activity of the neuron, and then you present the sound from different locations in space and see this neuron likes which location. Make sense? So you move the speaker at different locations, and at one point you will see that the neuron is firing like crazy, very high frequency. That means that is the preferred location for that neuron. That means that neuron processes or receives processes the signal when the source is at that particular position in space. Then he recorded another neuron, and this neuron prefers a different location. different azimuth, different elevation. Okay, and then another. So this way he recorded the neurons, um, at from different areas in the owl's auditory brain, and what he found that very systematically the neurons are located, number one, in terms of space. So a beautiful space map is present. Like this one likes one location, this one likes just another location but close to each other, this one likes slightly different location. In this way he created a beautiful map. It is a very time consuming experiment, extremely challenging. However, he very successfully performed this experiment and found that in the owl's brain that I mentioned at the very beginning, you have a space map which is not present in human or other creatures, where you have the neurons that can immediately detect the location, like this much degree elevation, this much degree of azimuth. Okay, and when this neuron is activated, your brain will know this is the location, one location. When this neuron is activated, your brain will immediately know that the location is here. Okay, so in that way the brain can quickly determine the location in space because of the neurons in the space map. Okay. So you see, this is a nice way to do the experiment during that time when we didn't have access or the programs to produce the sound at different time differences, uh, and he very successfully performed many experiments. And he also found that in the superior olivary nucleus, which is the area you must remember when we talked about the ascending pathway. This is the cochlea. So from the cochlea, cochlear nerve, particularly type 1 cochlear nerve fibers, type 1 cochlear nerve fibers take the signal from the inner hair cells through the ganglion cells. So this inner hair cells are here inside the cochlea, right? From the inner hair cells, type 1 fibers of ganglion neuron or cell will take the signal where? First to the cochlear nucleus. Cochlear nucleus. And you have cochlear nucleus in both sides of the brainstem, right? Then next is the olivary, superior olivary nucleus. Okay, and here a significant crossover takes place. Okay, and before the signal goes to the olivary nucleus, some signal will go through the trapezoid nucleus. Most of the signal will directly go to the contralateral olivary nucleus. Okay, small amount of fibers will go to the same side. So I am showing by thick line. Most of the fibers will cross over, will go to the opposite sides olivary nucleus. Okay, some will go through the trapezoid nucleus to the olivary nucleus of the contralateral side. Same thing here. Strong signal will go to the opposite sides olivary nucleus. Okay, and then you know that the signal will go through the bundle of fibers. These bundles are called the lemniscus. Lateral lemniscus is the bundle of fibers, and then the signal will go to the inferior colliculus. Inferior colliculus. Okay, and the neurons of the inferior colliculus will receive the signal, and then the thalamus, medial geniculate nucleus of the thalamus. So medial geniculate body or nucleus, and here some crossover take place. However, most of the fibers will stay same side, but some crossover takes place here. Okay, so medial geniculate nucleus of the thalamus. So you see that from your right ear, most of the signals are going to the left side, and from your left, most of the signals are going to the right side. Okay, and then you have the auditory cortex. From the thalamus, the signal will go to the auditory cortex. And if you see the structure of the auditory cortex, okay, so in the auditory cortex, you have actually a center core area, and this is the core area, and it is divided into three sub areas. Okay, three distinct areas where the neurons are positioned from high to low or low to high. That means in systematic order or systematic ways. So if you see this part, this is the primary auditory cortex, or A1. So this is A1, which is the primary auditory cortex, and this middle core portion is called the rostral core. Rostral core area, and this part is called the rostrotemporal core area. Rostrotemporal core. So in the center part of the auditory cortex, you have the core zone or area, and it has three sub areas, A1, rostral core, and rostrotemporal core. And as I mentioned, in all these three separate areas, you have frequency neurons are present in frequency maps. Okay, like low frequency neurons are here, then gradually high. So here is the highest, here is the lowest. Also in rostral core, you have opposite. Here you have the low, and this way is the high. Okay, and in A1, this way is the low, so start from low and then high frequency. Okay, so like 20 hertz to 20 kilohertz. That's the frequency range of human brain, right? So in owl it is different, but that's how. And then around the central core area, this surrounding area is called the belt, and outer shell is the parabelt area. Okay, parabelt area, and from the core area, the neurons of belt and parabelt areas get the signal. Okay, so first signal will arrive in the core area, will be processed there, then will be given to the belt area and parabelt areas. Okay, another thing in this connection, just know that the auditory cortex has the neurons systematically organized. That is the cochleotopic map. We say cochleotopic map, that means the frequency map. And if you record the neurons from the auditory cortex, the neurons have different tunings. That means some neurons are tuned very sharply. Some neurons are tuned widely. Some neurons are tuned in between, like tuning curve is intermediate. So this is sharp wide and intermediate. Okay, and as I told you that different neurons have different frequency preferences. That is called the characteristic frequency, or CF, also known as best frequency, or BF. Okay, that means that neuron prefers that particular frequency the best. Okay, so if you see narrow tuning neuron, this way is frequency. Okay, so this is low frequency, this is high frequency here. So for example, this is 10 kilohertz, this is 15, this is 20. So I'll say that this neuron's best frequency or characteristic frequency is 15 kilohertz. Make sense? And when you move a little bit away from that 15 kilohertz, like if you go down to 14 or 12 or 13 and go up to 16, 17, this neuron will not respond that strongly. Okay, very weak. So it will only respond very strongly when the frequency of the stimulus is 15. Narrow frequency tuning curve. This one is more wide. So if this is 20 and this is 15, this is 10, you see this one responds to like 12, 13, 14, 15, all the way to, I would say, 18, right? So when we'll present the stimulus at 12 kilohertz or 13 or 14, 15, all the way to 18, it will respond strongly. That means its characteristic frequency or best frequency is wide. The tuning curve is wide. Makes sense? And this one is much wider, more wider. Okay, so narrowly tuned neurons and widely tuned neurons, as well as intermediate tuned neurons we see in the auditory cortex. Okay, so sharp tuning is important to detect the tone or sound very precisely. That makes sense, right? So this is how you can also perform the experiment. Dr. Konishi also used camera, fixed the mini camera on the head, or a source, or you can also put a light, laser light on the head, fix it on the skull, and that will help you to determine in the dark room, because these experiments must be done in the dark room, so you can only study the sound, not the visual effect. So in the very dark room, how you will know which way the owl turned the head. One way is he placed or fixed laser light on the head. So this is the owl sitting here, this is the owl, and you put that laser light here on the head, and then I showed you how he presented the sound through the speaker at different locations in space, right, using that curved U-shaped rod and movable speaker. So you can present the sound from all different locations and see which way the owl moves his head, because owl will always move the head towards the sound source. This is the nature of the owl. Okay, so if this speaker is here and sound comes from here, the owl will quickly move his head. How will know? By looking at the light. When the owl moves the head, the light also moves, right? So you record using video camera where the light was in that dark room during that stimulus, and that way you can study the head turning which indicates the ability to localize the sound. Okay, if you present from here, then owl will move up. Okay, so the light will fall here on the wall, and you can analyze those video recorded light locations after the experiment, right? Okay, so as we know that our 2 ears help us to localize the sound, and just a few minutes ago I told you that superior olive is the area that receives signal from mainly from the contralateral cochlear nucleus, right? So superior olive has 2 parts, lateral and medial superior olive. Okay, uh, lateral superior olive neurons. So the neurons of lateral superior olive prefers high frequency sound. High frequency. They're sensitive to high frequency, and medial superior olive neurons prefer low frequency sound. And there is a significance of that. Okay, so medial superior olive neurons prefer low frequency sound, and that helps them to measure the time difference, interaural time difference, or ITD. And high frequency neurons of lateral superior olive mainly helps to determine the pressure difference or level difference, or sound pressure or loudness difference. So 2 different things, right? And both are useful for the brain to determine the location of the sound, interaural time difference as well as interaural intensity or level difference. Now the question is why the high frequency lateral superior olive neurons are more useful for interaural level difference. You see here there is a term that is called decay of sound, like high frequency sounds. If the sound is high frequency sound, when they travel, the pressure of high frequency sound gets much lower than the pressure of low frequency sound. Okay, so the intensity declines a lot here in case of high frequency, and that is something the lateral superior olivary neurons record. So since they prefer high frequency sound, when the high frequency sound comes, this is the owl head, or these are the ears. Okay, so if the sound source is far, then what will happen, you see, this is the sound source and the sound frequency is very high. When it will arrive here or here, it will lose the pressure or intensity more than if the frequency is low. If this is another sound source and if the frequency is low, that means the waves are longer, right? So this one, low frequency, will lose very little pressure or intensity or loudness, but this one, high frequency, will lose a lot more. So the neurons sensitive to this high frequency sound will get 2 different intensities, which are much more different than the neurons are receiving the sound um, from the low frequency source. Make sense? Since the loss will be more here, so the difference will be more, and that can be detected easily by LSO. Anyway, so the superior olive has two areas, medial superior olive, which is mainly processing the time difference, and lateral superior olive is mainly processing the intensity difference. And the time difference as well as the intensity difference both are very helpful to localize the sound. And if you see here cochlear nucleus, this is the section from pons just above the medulla and this area, and you see cochlear nucleus bilateral, right, that I mentioned. And from cochlear nucleus, remember I mentioned that most of the fibers will go to the opposite sides, olivary nucleus. Okay, some will go to the same side. Okay, and in the olivary nucleus you have lateral superior olive and medial superior olive. And also there is another area that is called MNTB, medial nucleus of trapezoid body. I mentioned the trapezoid body before. It is just before the superior olive, and trapezoid body actually inhibits the sound that passes through it. So the sound waves that or the signal passes through the trapezoid body is inhibited because of the presence of inhibitory interneurons in the trapezoid body. Okay. Now this is something very interesting. You see in the olivary nucleus you have the coincident detector. Okay, and coincident detectors are present in the MSO, medial superior olive, where the interval time difference is detected, right, not the intensity. So now you see this is a hypothesis based on the experiments and the response of the neurons of that area, medial superior olive. So in the medial superior olive you have the neurons. Those are termed as coincident detectors, and it has been hypothesized that these neurons will only fire when the time difference is precisely or at specific time differences. Okay, now let's see this one in condition A. Sound source is here, just in the midline, and as I mentioned, if the sound source is in the midline, that means the distance of your right and left ears are same. Distance is same. Okay, so if you have just these 5 neurons in the superior olive, 5 coincidence detectors, since the sound waves will arrive at the same time, when they will travel, you see, first the sound from this ear will go to this detector. Okay, and when from this one sound will arrive here, from this ear sound will arrive here at the same time, right? So 2 detectors are receiving sound from 2 ears. Make sense? And then next the sound will arrive here. At the same time, since the distance is same, sound will arrive here. So two separate detectors are getting the signal, right? One in the left side, one in the right side. However, when the sound will arrive here in this coincidence detector neuron, at the same time sound will arrive here because the distance is same, right? So at the same time, this is the event when at the same time from 2 ears the sound will arrive here. So this neuron will fire, makes sense? And that will be analyzed by your brain which neuron is being fired. If this neuron, which is sitting in the middle, is fired, that means the sound source is in the midline, same distance from your right and left ears. Okay, now let's see condition B. Now the sound source is more towards your right ear, right? So the distance of right ear is less than the distance of left. So what will happen is, see, sound will arrive here after the sound arrives here. So first the sound will arrive here. Okay, so when the sound will travel towards the brain, you see here the distance from here to here and here to here is same. That means since the sound arrived in the right ear before the left, so it will be able to travel more inside the brain. Okay, and since this one in left ear the sound arrived slightly later, what will happen? It will be able to travel less inside the brain or inside the auditory system. So at the same time, this coincident detector, that means this one, will receive the signal. Make sense? And if this one is activated, that will tell you, the brain, that sound is in the right side. If this one is activated, sound is in the left side, same distance in the left side. If this one is activated, sound is in the midline. Okay. If this neuron is activated, sound is more towards right. If this one is activated, sound is more towards left. Okay. So which coincident detector is being activated, that will give the brain the information that where the source of the sound